Good morning, good afternoon, and good evening, everyone. My name is Ron Vale, and I'd like to welcome you to Life Science Across the Globe. We're now in our third round of talks, and it's a great pleasure to travel to India and welcome our sister institute, the National Center for Biological Sciences. I myself have had the great pleasure of spending 9 months on sabbatical at NCBS, and it was a really, truly fantastic experience. And it's kept me in contact with NCBS and India ever since. We've heard 2 great rounds of talks from NCBS, and I'm looking forward to today's talks as well. And now I'd like to introduce you to the director of NCBS, Professor Jitu Mayer. Professor Mayer is a world-renowned scientist in cell biology and biophysics, and he's won many awards, and he's also a foreign member of the U.S. National Academy of Sciences. So I'd like to welcome Jitu, and he will introduce today's speakers. Jitu.
Thanks, Ron, for that kind introduction. It's also, great pleasure and privilege to be part of this Life Science Across the Globe series. And good morning, good afternoon, and good evening, all. Today's talks from the Bangalore Life Science Cluster, more than NCBS, are about 2 kinds of archives. The science talk is about building up a biological archive linked to understanding diseases of the brain. This is a multi-institutional effort involving our clinical collaborators from across the city, the National Institute of Mental Health and Neuroscience, and our sister institute located on our own campus, the Institute for Stem Cell and Regenerative Science, Stem Cell Science and Regenerative Medicine, that's inStem. We will hear about how this archive has been constructed from Raghu Padinjat, one of the leaders of this program. Raghu is a cell and developmental biologist and a professor at NCBS. He trained initially in clinical medicine and then obtained his PhD in molecular biology also at the NCBS. And after following this, he has established himself as a leader in understanding phosphoinositide biology and studying its role in the spatial organization of biochemical reactions and signaling. Now he's interested in using this archive for setting up to discover what he believes are how these processes that he's interested in affect human behavior and function. Following his talk, we have Bhavana, who is one of our newest faculty members on the campus at inStem. Bhavana's research area is to understand molecular mechanisms that regulate the development of the cerebral cortex in health and disease. She trained in Professor Shubha Tole's laboratory at TIFR in Bombay, in developmental mechanisms involved in brain, before she went to work in Professor Adrian Isaac's lab at the UK Dementia Research Institute, focusing on understanding the disease mechanisms in developing brain. She was attracted to our campus by this program, and you will hear why shortly.
Thank you, Jitu, for that introduction. And good evening, good morning, and good afternoon to everybody wherever they are. The title of my talk today is Discovery Biology for Neuropsychiatric Syndromes. And the topic of what we're going to discuss is how function emerges in a nervous system. As many of you will know, the final output of the nervous system, or any nervous system, is behavior. And this output arises through the physiology of the myriad cell types that underlie its structure. These cells are then put together to form circuits, and the circuits themselves are constructed of individual cells. Now, there are many levels and ways in which one can analyze the function of the nervous system. And to do this, there are a range of animal models that could be used. One could, for example, use a relatively simple model organism, such as the worm. And for all of its sophistication, it has only 300 or so neurons. And this has been an influential model in uncovering many principles of how the brain works. There are also a number of increasingly sophisticated brains that are seen in classical classically used model organisms such as the fly, the fish, and the mouse, which each in their own way have contributed to our understanding of how the brain functions. But today, what I'm going to talk to you about is how to understand the function of the nervous system by studying the human brain, which by far is the largest and most complex that we know of. There are, of course, logistical and other difficult problems in understanding the human brain and experimentally addressing how it works. But the history of understanding how the brain functions has a long history in studying patients. The person shown over here is Pierre Paul Broca. He was a physician who studied stroke patients who had speech loss. And from his analysis of these patients emerged the idea that there was this part of the motor cortex that was dedicated to speech, which we clinicians often nowadays refer to as the Broca's area. There was also the influential neurosurgeon Wilder Penfield, who lived 100 years later. And by carrying out live stimulations of the cortex during neurosurgical procedures, he was able to map in quite substantial detail which part of the cerebral cortex controls which part of the human body. And from these studies emerged an integrated view of this homunculus, which tells you the topography of the neurons in the cerebral cortex and what parts of the body they control. Today, of course, I'm going to talk to you about something somewhat different, but still related to the brain, and that is the problem of psychiatric syndromes. These are defined as illnesses that interfere with the way a person behaves, interacts with others, and functions in normal life. A bunch of fairly well-known disorders are included here, including mood disorders, addiction, schizophrenia, personality and anxiety disorders, and autism. These are a substantial medical problem. 2 to 3% of the population across the world is at risk. Here in India, where we are, about 193 million people, that's about 14.3% of the population, are affected by some form of mental illness. And depression and anxiety on the one hand, and intellectual disability of unknown origin on the other, are the most common. These are diseases which by their very nature have a prolonged course, and hence the treatment is also something that lasts for many, many years. The etiology of psychiatric syndromes has been investigated for a very long time. It's been known for a long time that there is a genetic component to the etiology of mental illness, and there are individuals and families which appear to be more susceptible to mental illness than others. Equally, it is now well understood that there are also environmental factors that influence the function of the brain. A number of perinatal and postnatal environmental factors listed over here can regulate gene expression, and through this mechanism can alter the brain, leading to disease susceptibility. So what are the modern challenges in dealing with psychiatric diseases? Well, we need to define better who are the individuals who are at risk. Since many of the diseases have overlapping syndromes, there is a need to identify or define disease types better. Who will become better and who will become worse as time progresses, predicting disease progression. And finally, treatment. We need to have the predictive power to understand which individuals will respond to which types of medication. Of course, it would be lovely to have novel compounds that help us to treat these diseases better, and also how to use existing therapeutics, in other words, repurpose them for controlling these diseases. The answers to these questions could come from basic neuroscience. If we could get a handle on the genetic basis of mental illness, the neurodevelopmental mechanisms that underlie how the brain is formed and functions, and how this responds or changes in response to environmental factors, and the cellular and molecular principles underlying this plasticity. So if you want to do all of this, what we need is a study that captures the evolution in brain structure, physiology, and function. It should allow us to identify the underlying genetic and environmental contributions and study the cellular basis of disease development. And to do this, we collaborated with our colleagues across the city in Bangalore at the National Institute for Mental Health and Neurosciences, which is a leading tertiary care hospital for the neurosciences. Over 500,000 people visit this hospital every year, and it has an adult psychiatry department, for example, which sees up to 100 new consultations every day and 200 to 300 follow-up consultations as well. And there are a number of specialty clinics dealing with each of the disorders that I mentioned before that have served as a location at which we've been able to recruit what are called clinically dense families. This is a pedigree of a clinically dense family. As you can see from the shaded symbols, there are a number of individuals that are affected by the particular disease that affects this family, that is bipolar disorder. You can see that these individuals are distributed across multiple generations. And indeed, even within a single generation, there are multiple individuals that have the illness shown in green, and this sibling, who is shown in blue, that doesn't have the illness. One of the reasons for choosing such families and studying this population is a study that was published a few years ago from David Reich and Dr. Thangaraj at the CCMB in Hyderabad. And what this study showed is summarized on the right-hand side of my slide. What is shown on the y-axis are individual population groups. And on the x-axis is an index of inheritance by descent, which is a measure of the inheritance of large segments of DNA between 3 to 20 centimorgans over more than 100 generations. What is shown in red over here, is marked in red, is the Finnish population and the Ashkenazi Jews, both of whom have been influential populations for discovering the biology underlying human diseases. As you can see, a number of the populations in the southern part of India show substantially greater inheritance by descent scores relative to the Finnish population, which is 1. And as a result, we believe that the study of these genetically dense families is likely to be very helpful in understanding the genetic basis of mental illness. So 250 families is the goal we are working towards, 50 each of the disorders listed over here. And for each family, we seek to recruit 4 individuals, the proband, a second patient, and 2 unaffected individuals, 1 above the age of risk and 1 below the age of risk. And each individual in these families is phenotyped with a great deal of detail at the level of behavior, at the level of brain structure using structural MRI, and at the level of brain activity using functional MRI and clinical electrophysiology. This serial assessment this is not a one-time assessment, but a serial assessment of patients, at-risk individuals and controls at 2-year intervals over a 20-year period. And all of this information is being archived in a digital database to facilitate research, both now and hopefully well into the future. So of course, all of these types of clinical neuroscience that I just mentioned in the last 2 slides will inform us about changes in behavior, physiology, and likely circuits. But what it does not allow us to do is understand what are the changes in the brain cells that underlie human brain mental illness. For example, one would like to understand what are the changes in synapse development in the brain when a person is ill. What are the changes in synapse chemistry related to neurotransmitter production, receptors and metabolic changes? And finally, what are the changes in neuronal function? To do this, what we've done is to go about creating a repository of stem cell lines from the individuals, the families that I just mentioned. And so starting with a sample of blood, we go through several steps to generate induced pluripotent stem cell lines and neural stem cell lines. And these are being bioarchived in a biorepository. These stem cell lines can also be differentiated using the principles of developmental neurobiology that have been discovered with hard work over many, many years. And as you can see in this slide, you start with neural stem cells. And as the culture is maintained in culture for over a period of 3 months, you progressively see the development of immature neuron markers, mature neuron markers, known cerebral cortical markers, and finally, the development of synaptic markers and enzymes and proteins involved in the control of neurotransmission. We can use these types of cultures to understand what is the altered cell biology in these patients and what is the physiological properties of these neurons. And here's an example of one such culture, which has been differentiated in vitro. And you can see the neurons, which are labeled in green, and the glial cells, which are labeled in red. One can load up such cultures with fluorescent indicators. We can monitor electrical activity. And if I play this video, what you will see is the flashes of fluorescence depicting the activity of the neurons as they develop in the dish. And using such cultures, one can monitor, for example, the progressive increase in transients as a function of age in vitro, as well as the change in the properties of these transients as the culture matures. Whatever we discover in these studies, one would want to know what is the basis of this phenotypic variation. This likely arises from two factors, either environmental inputs that influence brain function, and this will be derived from the detailed clinical characterization that my colleagues at NIMHANS are carrying out. The other possibility, of course, and the great interest, is the genetic differences that might give rise to these phenotypes. And for that, what we've been doing is a systematic analysis, exome sequencing and analysis from individual families within this cohort. And here is an example of one such family with BPAD. What you can see in red are linkage regions which have previously been linked to these mental illness. What you can see in blue are well-known loci and genes for BPAD. And what is shown in green are the loci that have been derived from the analysis of this family. And we are progressively trying to look at each of these and try to work out what impact they have on cellular function in the brain using the experiments I just described. So in summary, then, I've told you about three broad types of data that are being collected. A cohort of clinically dense families with very detailed phenotyping in the clinic, a biobank of iPSCs that can be differentiated to study cell biology at various levels, and genome analysis of the same families that allows you to relate what changes might result in the phenotypes one observes. Viewed from the perspective of a single family, this might look as follows. What you can see is the same family I showed you before. And for this individual, for example, over here, we have a number of resources that we have collected. The endophenotype, which is from the clinical characterization, the iPSC and NSE that I just described to you, and the exome sequence. And by using these, it is possible, for example, to compare all of these individuals in orange that are affected by BPAD with their sibling in green from the same generation who does not have the illness. Alternatively, for example, one may compare two individuals who are affected by the illness, one in green that is drug sensitive, and the other one in red, who is not sensitive to the same treatment. Over the years, the repository has collected a large amount of information that is summarized on this slide in the different categories. And you can get an idea of what is present in this biorepository by visiting our website, which is shown here at the bottom of the slide. If you go into this repository, you will be able to see all of the elements, individuals and their families that are listed over here. It's possible to sort this information by the disorder that you're interested in, or by the availability of an iPSC for that particular individual, or by, for example, the availability of exome sequence, or indeed by any of the clinical features that you might be interested in. So a project of this scale and magnitude could not be done without collaborative work of a large number of colleagues at our three institutions, and I've listed all the principal investigators that are involved in this project. But equally, we've had over 100 staff work on this project over the years, trying to build up this resource. And most importantly, for patients and their families, without whom this project would not be possible. This research was supported by the Department of Biotechnology, the Government of India, the Pratiksha Trust, and by NCBS and inStem. With that, I thank you for your attention, and we'll take questions later. Now I'll hand over to my colleague, Dr. Bhavana Muralidharan, who will describe her interest in cortical development and link it to how she's going to use this resource to answer some questions in that area. Thank you.
Thank you, Raghu, and thank you, Jitu, for the warm introduction. Good morning, good afternoon, and good evening, everybody. One of the regions in the brain which is majorly affected in these neuropsychiatric syndromes is the cerebral cortex, which is the seat of higher order functions in the brain. A hallmark feature of the development of the cerebral cortex is the various neural cell types in the cerebral cortex. They all come from a common pool of stem cells, which are called progenitors. And chromatin regulation, which is the interaction between the chromatin factors and transcription factors, which then go on to regulate the gene expression, plays a fundamental role in generating the diverse neurons and glia which we see in the cerebral cortex. Indeed, it's not a surprise that chromatin mutations in these chromatin modifiers, which lead to chromatin misregulation, is thought to be at the root of neurodevelopmental disorders. The generation of the laminar, the 6 layers of the cerebral cortex, begins with these progenitors dividing first to generate the layer 6 neurons, marked here with a transcription factor TBR1. Then the same progenitors go divide and generate the layer 5 neurons, which migrate past these layer 6 neurons and sit on top of that, marked here with FESF2. And so on and so forth, stepwise, every layer is generated till all the 6 layers of the cerebral cortex are generated, marked here. The upper layers are marked here with CUX2 and ROR beta. A few years back in my postdoc, I studied the regulation between the switch between the layer 6 neuron production to the layer 5 neuron production by studying the interaction of a transcription factor LHX2 with the chromatin modifiers from a repressor complex known as the nucleosome remodeling complex, whose function is to remove active marks from the enhancers and transcription start site of target genes and thereby keeping the target genes repressed. In this case, this interaction between the transcription factor and the members of the NUT complex resulted in keeping active marks at low levels on 2 target genes, FESF2 and SOX11, and keeping their levels low. These 2 target genes are very important genes, important for the production of layer 5 neurons. And this interaction kept the levels of FESF2 and SOX11 repressed at the time when layer 6 neurogenesis was underway in the cerebral cortex. When LHX2 was removed, we saw that these chromatin modifiers could not access either the enhancer or the transcription start site, which then led to an increase in active marks at these the loci of FESF2 and SOX11, led to an upregulation of FESF2 SOX11. And the consequence of this was that in an LHX2 mutant, the layer 5 was expanded at the expense of layer 6 neurons. Indeed, the mouse is a very valuable model system to study conserved mechanisms, but shown here is a key and important difference between human and mouse cortical development. The expansion of a zone, which consists of intermediate progenitors, which is called the outer SVZ, and it is thought to contribute to the neocortical expansion, which we see in humans, which actually results in a large brain which is folded into gyri and sulci as seen in this cross section. Thus, this creates the case for having a human-specific model system to study human cortical development and human-specific disease mechanisms. Thus, cerebral organoids, which are derived from iPSCs, are not just valuable to study human brain development, but given that these come from patient-specific tissue, these are very important to study disease pathology to understand patient-specific genetics of the disease. My own lab's interests are to understand the molecular and cellular mechanisms of the neuropsychiatric disorder schizophrenia, which is characterized by aberrant perceptions, thought, and social connectivity. Family studies show that it is heritable and that it is neurodevelopmental in origin. There are many studies which are from Caucasian populations and very recently from African populations as well. But there are no such studies from the Indian populations. Thus, in this context of my own research interests, the AD-based program is a very valuable resource for investigators like me who wish to do human in vitro disease modeling of psychiatric disorders. Firstly, this resource, this program has many clinically dense families, several of whom have schizophrenia. Secondly, the exome sequencing from these families, which has been performed, enables us for the identification of putative risk variants which could be contributing to the disease. And thirdly, there exists parallelly from these patients clinical assessments to match the cellular and molecular studies which researchers like me wish to undertake. Shown here is a family in which several members seen here in the shade, seen here in shaded boxes and circles. Several members of the family have schizophrenia and bipolar disorder, not just in one generation, but running across generations. And also there are several controls in this family as well. The exome sequencing of this family has been performed. And of the several putative risk variants which have been identified, one such risk variant which really interests me, given my own background in understanding chromatin regulation and cortical development, is a risk variant in the epigenetic modifier SETD6. Now, all of these family members have undergone rigorous clinical assessments on several parameters, as mentioned here, which Raghu also mentioned in his talk. And the risk variant, putative risk variant, which interests me is is in the epigenetic modifier SETD6, which methylates histones. And this putative risk variant has been seen only in affected individuals, but not in the control. We wish to now undertake molecular and cellular studies by creating cerebral organoids from these iPSCs derived from these patients, and then ask the question that how may this putative risk variant in this epigenetic modifier SETD6 lead to cell type specific chromatin accessibility changes in these patients, which could then lead to altered epigenetic landscape and altered gene expression signatures, which may then be seen as altered neural development phenotypes, which we would like to capture in cellular assays. We would like to perform cellular assays to understand which step in neurodevelopment is affected. There could be defects in progenitor proliferation, there could be defects in subtype specification, or there could be defects in migration, dendritic development, all the way to synapse formation. And we wish to understand what are some of these neurodevelopmental phenotypes which we can see from these patient samples. Thus, the AD-based resource is a very valuable resource for starting investigators like me who wish to understand the emerging function of the nervous system, development, which will start from molecular changes, which could be mutations, genetic mutations, or altered epigenetic mechanisms, which then lead to targeted gene repression or activation, which then leads to altered neural development and going all the way leading to a dysfunctional brain. And all this we would like to understand, my lab would like to understand in a human-specific disease mechanism context. This brings me to the end of my talk, and I would like to thank my postdoc mentor, Shubha, and collaborator, Sanjeev, and all the funding agencies, and I'm happy to take questions now.
Well, thank you, Raghu and Bhavana. A database, an archive for the genetic basis of neuropsychiatric syndromes is a very useful resource indeed. And thanks to both of you for the great talk and for introducing the different aspects of this database to us. So moving on to discussion, let's start with a technical question. So psychiatric disorders present themselves on a scale of severity, right? You have mild, moderate, severe, and places in between. So do you pick a certain scale of severity when you identify your patients? And if so, how consistent is that across cases?
As I mentioned, the patients and family who are recruited into this study are assessed clinically by my colleagues at NIMHANS. And in doing so, they do get assessed in a great level of detail and classified using standardized clinical nomenclature that is used to be able to objectively identify a patient as being of a particular type of disorder and at a particular stage of it. And these are, for example, things like the DSM and the ICD classification. So the reason why one does this is that it should not be the case, to the extent possible, that individuals working in different parts of the country and in different parts of the world do not classify patients in subjective ways, but rather in an objective way that allows anyone who is looking at the data to be able to compare it with what is being done elsewhere. So the short answer to your question is that standardized systems of classification are used to assess the patient at the point of entry into the cohort. The second part of the answer is that there will, of course, be unaffected individuals in that family. If they are below the age of risk, then it is possible that they are normal now and they may develop the illness going into the future. And as the serial assessments take place, one will be able to watch the earlier stages of the illness developing and progressing. And this will also be quantified using these standardized scales that psychiatrists, not just here, but all over the world use.
All right. Thank you. Thank you for the answer. We now move on to audience question. We have a question from Shiva. Shiva, would you like to ask your question? If so, please unmute yourself. All right, I think I'll go ahead and read out the question. Shiva's question is, to what extent the environmental factors and modern lifestyles have impacted these mutations? And if so, are these transmitted through generations?
I mean, I think that it's probably fairly well understood, and indeed is a matter of great interest as to what the impact of the environment is on the developing brain and its function. The human brain is one of the few examples in which a substantial part of development takes place ex utero, that is after one is born, and all the way up to adolescence. The brain is in a state of development with synaptic pruning, reformation of synapses, and so on. And naturally, therefore, environmental factors that the person is exposed to will have an influence or could have an influence on the function of the brain. As I mentioned, the first step that happens after a patient is recruited into the study is that a detailed clinical history and assessment is undertaken. And I think about 75 individual fields of information relating to clinical history are collected by my colleagues in NIMHANS. The purpose of this is precisely this, to quantify what are the factors from the environmental exposure of a person, be it social, economic, and so on, that might have contributed towards their illness. The genetic analysis, on the other hand, is of course the template or the background on which environmental factors may work. And therefore, I would say that you need a combination of both of these tools, the clinical characterization of the environment, the clinical documentation, I would say, of the environmental factors, plus the genetic background, to be able to arrive at a consensus on how what combination of these two things contribute to mental illness. I think the jury is probably out yet on the matter of what is a relative contribution, but I think generally everybody believes that it is a combination of genetics and environment.
That's true. That's probably right. The combination of genetics and environment probably have an influence on these disease. All right. Another technical question here. So this is regarding exome sequencing. So exome sequencing is clearly the way to go to understand variations in coding sequences. But then what about differences or changes in intronic sequences? Because I would imagine regulatory elements to also have an influence, which is probably not something you would capture with exome sequencing strategy.
Yes, that's a very valid point. Exome sequencing, for the most part, will capture changes in coding sequence variants. And in the initial design of the study, this was linked to the fact that we were going to be looking for phenotypes in the iPSC-derived models. But what we do plan as we go forward is that for the most interesting families and for the most relevant cases, we will also supplement the exome sequencing with whole genome sequencing. And I think that should give the type of information that the question relates to. What are the intronic sequence variants or the intergenic variants that are likely to have an important contribution mainly to gene expression and therefore brain development and disease phenotypes. So I think this is something to look forward to in the future, and we do indeed plan to add that as we go along.
Right. Thank you. All right. Let's see. Here's a question from another question from the audience. Annabelle. Annabelle, would you like to ask your question? If so, please unmute yourself. Okay, I'm going to read out the question. Annabelle would like to know, how do you deal with still a healthy person enrolled in this kind of study who would eventually develop mental disorders at a later stage?
Well, all enrollment in the program is by informed consent. And typically, one is recruiting a family with both affected and unaffected individuals. They would be characterized at the point of entry. And if there are any not obvious clinical features, and this would, of course, be picked up by my clinical colleagues as they look at the patient in great detail. As we phenotype these individuals every third year, any of the earliest signs of mental illness at whatever level of brain function one is looking at, either behavioral, structural, or functional MRI or clinical electrophysiology, this would become evident. The question, of course, may be, I'm not sure whether the question is an ethical one or a scientific one, but if it is normally this is not an interventional study, and we would therefore, if a person who is well now develops the disease as we go along, they would be treated as per the existing protocols approved for treating neuropsychiatric illnesses. So this is not an interventional study, but an observational study.
All right, thank you. Bhavana, here's a question for you. Speaking of the epigenetic landscape, how much of the epigenetic memory is retained during the reprogramming of iPSCs?
That's a very valid question. And so several studies have seen that the marks naturally, once you derive these cortical neurons from the iPSCs, several of these marks do come up because there is some sort of a memory as you differentiate these stem cells into specific lineages, in this case, say cortical neurons. You do get some of these marks, and in fact, the chromatin accessibility has been studied, and it is very similar to what you would see during human fetal development. But having said that, of course, it's not a complete system because it's a reprogramming system. I would like to say that organoids is probably the best possible model system we have right now to study human-specific mechanisms, besides, of course, fetal tissue. So right. The answer is that, yes, many marks are conserved, but I cannot say for surety that it's true across the board or throughout the genome.
Fair enough. All right. Okay, so here's another question from the audience. Shiva, would you like to ask your question? Or would you like, please unmute yourself if you'd like to ask. Okay.
Hi. Hello.
Go ahead.
Actually, it's a great talk. I would like to know, does telomere length also decreased in brain organoid cultures upon culturing for a long time? If it's yes, how it is related to human situation?
Okay, so Shiva, thank you so much for this question. So pioneers in the field of cerebral organoids, like from the lab of Madeline Lancaster, Seju Pasca, and Agil Teagstein, have kept these organoids even say up till 300 days in culture. And I'm not aware of any such study where they've looked at telomere decrease. But I think 300 days of a human cerebral organoid is still just perhaps, gone through development and just become postnatal or just become just like a adolescent. So the shortening which you are describing, maybe it's really in aged neurons or aged cultures. So I'm not aware of any study where such has been studied in the normally forming organoids, but people have induced aged cultures by overexpressing progeria and some telomere inhibitors, but that's just specifically to look at aging disorders.
Okay, thank you.
Yeah. Thank you, Shiva. So thank you, Raghu and Bhavana, for the great talk and discussion. And with that, we will move on to our next speaker, Venkat Srinivasan, who will talk to us about yet another archive,